We talk about sensing and we talk about we want to measure the presence of specific biological entities. But how do we define a sensor? There can be different ways how we sense. So there are three parts of a sensor basically. You have molecular recognition where we want to look for very specific things. So these triangles or circles or hexagons, so they're basically depicting different molecules. We are interested in finding one molecule than the other. So what we would do is we would put some things on the surface that can bind to those molecules. Now, once those molecules bind, we want to design some change that we can measure. That change can be the chemical change, can be the weight, can be a light, can be sound, heat, bunch of changes that we can measure. But the change then most of the times has to be, it happens with some devices like electrodes or microbalance or photomultiplier or photodiodes or thermistors. So there are many different uh, systems that can measure those changes. And at the end of the day, you want a readout, you want an electrical signal that comes out. Can you give me an example of a biosensor that you know? There's, there are glucose sensors all around us. There are pregnancy test kits. There's cholesterol test kits. There's a whole bunch of things that are used either at home or in industry, right? We have gas sensors. We have ion sensors. If you work in an industry, if you make something, if there are chemicals around, we have a bunch of sensors. What are athletes worried about? Athletes. What kind of testing do they do on athletes?
Steroids.
Steroids, right? Urine test, right? So they're looking for specific chemicals in there. They have basically test strips. They simply test strips, they dip it and they see the color change because that they have specific chemicals over there. If they find those chemicals, their reactants, they would go maybe change color. Or for glucose meters, simply the blood sugar changes the resistance of a coil, a small wire in the test strip. The change in resistance tells you about the level of glucose calibration, the thing which I was talking about before. So, but what we are doing here, nano and MEMS, there are lots of new sensors always coming out. So they're integrating nanotechnology with new sensing to get new sensing capabilities. So a whole bunch of measurements for blood, for saliva, even so much so tears. There's all kind of bodily fluids that they're using to sense for specific things. For as simple as sugar to maybe cancer, to look for uh, brain diseases. A lot of it happens at small scale, 2 nanometer, but a lot of that happens at large scale as well. So these are some areas of applications where maybe uh, for diagnostics, where we want very, very specific detection of a biomarker. And we want to make sure we don't get false results, which comes in selectivity. Sensitivity relates to that we don't want to miss very small changes in those things. Another thing called nanomedicine or personalized medicine. Yeah, when we get headache, we take Tylenol, right? Or we get cramps in our leg, we get Tylenol. So it's okay to get Tylenol. Everybody gets Tylenol. But when we talk about very diseases which are very specific, like maybe a cancer in some part of the body, we cannot give all the drugs to all the patients. So drugs need to be made tailor-made for those people. So you maybe you know they create cocktail for uh, maybe cancer patient because their cancer molecule or their cancer cells would show certain properties. So they would then give the drug that attacks those properties. So that's the concept behind what we call nanomedicine. We want to, number one, make it very specific the person, so the cost of healthcare comes down, the the life becomes, it's not too, the side effects become less, so the personal life or the quality of life is not degraded too much. There are legal places where we need these sensors. There is a whole bunch of needs that go into making these sensors. We need better accuracy, higher accuracy. We want small sample. We want to detect for many things, maybe in one test. At the end of the day, it's the cost and time that dictates that we want to cut down cost and want to get results faster. Here's an example. On the right hand side, it's a big system. There's smaller system also. They call PCR machine or other processes in labs. On the left hand side, you see with the one dime, it's a PCR chip, same process, but now it can be done on a chip scale. So you don't need large sample to start with, right? We don't need too much energy to do that. And it's small, you can get results much faster. So essentially, there is a long range of lists that why we want to do this, why we want to create nanosensors for biosensing. This is my son. We all have got shots, right? So we all have gone through that, uh, some sort of invasive procedure which hurts, right? Or which makes discomfort. So that's where, again, if we need smaller sample, we can, we don't have to inject the whole needle. If we can come up with new ways, we can look at the samples that are easily available, right? Saliva, urine, uh, tears. Now, if we make those things, we learn from things like Human Genome Project, which have identified genes, right? So now we know what are the chemical or how would they come together. So we can create from that knowledge, then we can create these kind of devices, right? So I put bunch of these things here to sum up. So this is DNA microarray on the top left-hand side, right? So where we put all millions of drops for specific genes, and then you would run the sample in and you can find out. There are glucose sensors, there are all kind of things that we can measure, pregnancy test kits for ions, for drugs. But the lower level is a bigger scale. So you have to see that, yeah, we have MRI machine, X-rays, or implants, which are already effective, but there are many, many, many more diseases where we can learn from nature and make smaller things. So mimicking nature, if you think about it, lungs have a big pipe coming and then many smaller pipe, and they keep on becoming smaller and smaller until the oxygen, until the gas exchange happens, right? Oxygen gets into the blood, carbon dioxide gets out from blood and gets into oxygen. Can we mimic that process on the surface of chip? There are many people who have tried to do that. And this is a real picture showing that kind of approach. Can we identify problems at single cell level? So here's the picture of cell. We have seen that before. Can you make an interface where one cell lands here and you can measure changes at single cell level, right? Whether that's, again, chemical change, physical change, whatever problem happening at that scale. The lower left picture is a conformable sensor. So instead of having a sensor which gets on the desk, can we put it like a band-aid on ourselves? Recently, uh, we I came across they have got temperature sensor which is like a band-aid, right? Big deal. Temperature sensor, we all can look at temperature, right? Use thermometer. But think about people who are diabetics. They lose sensation in their feet or in their hand because of their neurons get destroyed because of high sugar level. Their feet are cold and they don't even feel their feet are cold. But putting that kind of patch in their feet can help us, because now your device or gadget can tell you your feet are exposed to high cold. So you need to put on maybe socks, or you need to put your feet, uh, protect your feet, because that's a major problem. Yeah. There is a huge number of people who get amputation, especially who are diabetics. So these kind of things can learn from nature and do something about it. This one on the right hand side, lower one, that's showing you a tissue, especially there's a part of human body called basement membrane. All the living things, they have it. If you look at it, it has nano texture to it. It has texture which can be created in the lab at the same scale. So essentially, there are so many directions where MEMS and nanotechnology can have an impact, and not just diagnostics, but therapy, tissue engineering, now what we call as regenerative medicine, creating organs for people who have problems with their existing organs. We are getting there. We'll talk about that also going forward. Cell biology, can we look at single cell levels and see how cell biology or cell problems are occurring there at that scale? Materials, where we learn from biology, right? So because bio, just look at your nails and you'll be amazed. We create this hard material, all of us. Do we have a machine that can create such a hard thing? Look at your teeth and you'll be amazed. It's hard material. We exist with all these hard materials. Can we recreate those materials by following the maybe something inspired from living systems? Bioelectronics, another area where we want to measure biological things. We talked about a few things today and we'll look more. Bioinspired fabrication, how nature creates things and can we create things in the same